All right, this is DNA, D for Daniel, A for
Aiden.
Yes. Let's do this. We're here to talk about cellular respiration.
Yeah.
We're trying to, get you guys, focused and make you guys more adapted in cellular respiration so that you guys can be better prepared when this topic comes into your class. All right, before we get into, the whole glycolysis, Krebs cycle, and oxidative phosphorylation, we're going to actually dive it into, like, the pre-stages is what I like to call it, of cellular respiration. One of these steps is that photosynthesis is another type of energy. This is symbiotic with cellular respiration. If you don't know what symbiotic means, it actually means that, that one cannot be there without the other. They both are needed in order to function.
It's mutual.
Mutual, yes, exactly. The oxygen produced from photosynthesis needs to be used in cellular respiration to produce carbon dioxide so that photosynthesis can take place, which is basically a whole circle of like things that you need for one so that the other one stays, and that's why plants are so important, and that's why it's so important to keep the ecosystem so safe.
For us humans and for organisms as well, animals.
Another key component of photosynthesis that just is really important is glucose. This is a very important formula in cellular respiration, and my friend here would explain it.
Yes, C6H12O6 is glucose, and 6O2 is oxygen, and both of those within cellular respiration yield 6H2O, which is water, and 6CO2, which is carbon dioxide, and it also produces ATP so that cellular respiration can take place.
Exactly right. This formula is also vice versa. Whenever you're gonna, for example, photosynthesis is the vice versa of cellular respiration, as I explained, and photosynthesis is the vice versa. Yeah, you get the point. The next thing that you need to know before going into the amazing world of cellular respiration is redox reactions. These are chemical reactions where electrons are transferred from one atom to another. And what this means is basically, I like to think about it, I remember very deeply about redox reactions when I took chemistry. It's basically for an electron, the electron number on one side of the equation switches when you after the yield sign. So for example, in this equation, the C in this equation is the thing that is being oxidized, I believe, or reduced. It doesn't matter which one. As long as one of the two things is going on, the equation is technically considered redox. So that's what basically a redox reaction is, and it relates to the cellular respiration formula. Another thing you need to understand before diving into cellular respiration are electron shuttles.
Yes, the main electron shuttles are NADH and FADH, and also before
they're oxidized,
it's NAD and FAD.
Yes, and we'll talk about when they are oxidized. Actually, right now, we're gonna move on to that was basically what you need to know before you start into the main concepts of cellular respiration.
Yes.
Now we're going to talk about glycolysis, which is, if I was gonna say steps, but actually Ms. Deschamps corrected us in this. Cellular respiration is an ongoing event. It's not in steps. Actually, everything is going at the same time. It's very key to remember that. It's really important to remember that all the things in cellular respiration are going at the same time. These are not steps. But for the purpose of understanding, we'll call it step one. Glycolysis. Glycolysis, would you like to explain this one?
Yeah, I'll do that. Glucose is it's two molecules that are converted to 2-pyruvate.
Yeah.
And then it requires 2 ATP, and it produces also 4 ATP. And 2 NAD
are produced into NADH.
Yeah, are produced into NADH.
This is what we were talking about, the oxidation. This is when NAD plus is oxidized into NADH. And a little bit more into what he was talking about, the 2 and the 4. You actually need 2 ATP in order to do glycolysis, and you produce 4, but you actually get a net gain of 2. And this is basically 2 per glucose as well. Would you like to explain anaerobic fermentation as well?
Yes, and in anaerobic fermentation, NADH is oxidized back to NAD. So in anaerobic fermentation, there's no oxygen within the anaerobic fermentation. This enables glycolysis to keep functioning.
Yes. Basically, the the NADH being oxidized back to NAD allows it to be basically to be reused in glycolysis, and this is only used whenever there is absolutely no oxygen, or little oxygen, I should say. Important ways of fermentation, I guess you could say, is the ethanol fermentation, which is pyruvate being converted to ethanol and molecule of CO2. Ethanol is just, another compound. It's kind of like, I think it's an alcohol, I believe.
It's like gas, no? Or...
It can be producing, basically, but that's the thing. It's one of the important ways of fermentation. Another way of fermentation is the lactic acid fermentation. This was actually on the AP Bio test. It was in the mussels, actually. Our mussel boy here. We took a question over the lactic acid fermentation. And that's basically pyruvate is being just converted into lactic acid. It's really simple, but it's on the test. It's very important to remember that this occurs in all cells. And if there's one thing you need to remember, is that everything does glycolysis, which ties back to evolution. And that's really important to know, because they will love to do this on the AP Bio test. They love to tie two different topics together, and that's one of the things that I think would be tested the most, which is glycolysis being tied to evolution, that both relate to each other, basically. And we're going to move on to the next step, which is
pre-Krebs cycle. So before the Krebs cycle, there's
while being transported into the mitochondria,
pyruvate is converted to acetyl group and one molecule of CO2.
The CO2 is released as a waste product.
Yes, and as the acetyl group is attached to a coenzyme, and this is the carbon input into the citric acid cycle.
Very true.
Also, NAD is reduced to NADH, as we mentioned before, and this is this occurs between the mitochondria and the cytoplasm.
Yes, this is actually, it's kind of like a mid segment between Krebs cycle and glycolysis. It's not really, it's the the name pre-Krebs cycle. It's basically before the Krebs cycle. Another thing that I would like to mention is that early in the video we actually got something wrong. We said that NAD is being oxidized into NADH, is actually being reduced. I'm very sorry for that mishappening. But yeah, actually when NAD is being reduced to NADH, and whenever it's NADH to NAD, it's being oxidized. I'm sorry for that mishap. But that's basically the pre-Krebs cycle. You need to understand that the pyruvate is attached to acetyl group, and then that acetyl group is attached to a CoA group. So it makes acetyl-CoA, which is what is needed in the Krebs cycle. And I will explain the Krebs cycle. The acetyl group is attached to oxaloate. I'm not sure how to pronounce that, actually. I have to have the word written down, because it's actually a really weird word. All I know is that it's attached to it, forming citric acid, and citric acid is really important in cellular respiration. This carbon from the acetyl-CoA is oxidized into CO2. This is really important because it's released as a waste product. 3 molecules of NAD plus are reduced into NADH. 1 molecule of FAD, remember NAD and FAD plus are electron carriers. It's very important to remember that. FAD is reduced into FADH2. It's really important to remember that too, because that's the only thing that, besides the F and the H, it's the only thing that's different between NAD and FAD. It's FADH2. 1 ATP is produced. This is probably the least amount of energy produced in all of cellular respiration. It's basically happens 2 times per glucose, this whole entire cycle. So 1 glucose for the 2 pyruvate happens twice. That's why there's 2 pyruvate. It would make sense. And this all occurs in the mitochondrial matrix, while glycolysis occurred in the cytoplasm. It is actually very important to understand that if you noticed that in glycolysis and Krebs cycle, almost little to no energy is produced. In glycolysis, it's only 2 net gain. In Krebs cycle, only 1 net gain is produced. So it's concerning that you don't produce that much energy, which is why we're going to move on to oxidative phosphorylation, the main source of ATP. Go ahead, sir.
So what's reduced is NADH and FADH, and they are oxidized into the electron transport chain.
Mm-hmm.
These are proteins located on the cristae, the inner mitochondrial membrane. Mitochondrial membrane, yes.
That basically is just saying, if you don't know what the electron transport chain, it's basically it's a row of proteins, I guess, on the cristae, which is, if you've ever seen, actually I think I have a picture here. I'll try to show the picture. It's right there. It's basically it's, if you've ever seen a mitochondria, you see the lines in it. That's the cristae. It's actually a really good icon that Ms. Desham showed us. Hopefully she'll show you guys. It's basically oxidative phosphorylation takes place on the cristae, is what I'm trying to get at. Go ahead.
Also, electrons flow through the electron transport chain.
Tongue twister there.
Through concentration gradients.
So what a concentration gradient is basically like, it's diffusion. It's another type of biology topic. It's basically going from like a high concentration to a low concentration, and this does not require energy, which is actually really important to understand, because if it did require energy, you wouldn't have that insane ATP net gain. And that's basically the electrons, the H plus ions that are getting diffused into the cristae.
Yes, and also when the electrons move through the ETC, the energy that they release is used to pump protons, H plus protons, from the matrix into the inner membrane.
That's basically what I just explained.
Yeah, basically.
Yeah.
And the oxygen is a final electron acceptor, and it creates H2O at the end of the ETC as a waste product.
Basically explains itself, like, as the O2, as you ever wonder why you need to breathe, this is why, I think, because if you breathe in the oxygen, it allows you to have that as the final, as it explained, as the final electron acceptor, and this would allow you to mix with the H protons, and it'll allow it to make H2O, which is why you ever wonder why, like, even though you haven't drank in water in, like, let's say, like, you haven't drank in a liquid in over like 8 hours, let's say, and you still need to pee. That's because of this, because cellular respiration produces water, which is something a lot of people do not know. So yeah, fun fact. Go ahead, keep going.
Oh, and those protons that we mentioned a while back that were pumped out into the inner membrane, they go back in through protein ATP synthase.
Yes, that is true. And
that right there produces ATP. Also,
that this process is known as chemiosmosis, which is what you basically just explained. Within the ETC, there's a proton gradient, which we've already explained. Protons go through the mitochondrial matrix through protein channels, and the protein channel that he just explained is ATP synthase. The only protein that can have, protein ATP synthase is the only proton gated channel on the cristae. And other than that,
and also we forgot to mention a little while back that the oxidized NADH and FADH are fed back into the Krebs cycle and glycolysis, so it could be reused.
That is very true. And whenever the positive protons, H plus protons, diffuse through ATP synthase, free energy is released through catalyzed. Basically, what that means is ADP, notice how I didn't say ATP, ADP gets another phosphate and produces ATP, which is energy, adenine triphosphate, as you said before.
Adenosine triphosphate.
Yes. And that's basically all that there is to cover about cellular respiration. You just have to remember glycolysis, cytoplasm, Krebs cycle, matrix, oxidative phosphorylation, cristae, and all the key components that FAD and NADH basically uphold in cellular respiration. It's very important to know like how the movement of NAD and FADA and FADH and NADH move through the cellular respiration. So I'm glad we could be of assistance in trying to help you with cellular respiration. Thank you for your time, and this is DNA signing out. Peace.